Welcome everyone to another edition of You and Me and Multiple Sclerosis. My name is Pam, and I've been living with multiple sclerosis for 37 years. I keep looking up more information about things that I'm interested in, and I'd like to share some of it with you because you might even find it interesting too. Recently, I was reading something or listening to something, I can't remember which, but the person mentioned that multiple sclerosis is essentially a form of accelerated aging. And that really intrigued me. So I decided I'd better look into that and see what they were talking about. Well, one thing I knew right off the bat was that it wasn't the kind of aging that we think of when we think of, well, I need to dye my hair because of the gray, or I need to use a certain face cream to hide my wrinkles. It has to do with the cellular level of our bodies. I went out to the internet and I looked up some of the research that has been done on the subject, and I found that there's quite a bit going on because, of course, aging in general is something that scientists are very interested in understanding. What are the mechanisms? How might we reverse it? How might we stop it? How long can we possibly live? That's been a big deal, but the recognition came pretty quickly that a lot of the neurodegenerative diseases like MS are actually very similar to aging at the cellular level. So I'm going to go ahead and show you some of the things that I found. With this particular field, it gets very technical very quickly. And I've had biology classes, and I am interested in science, and even I was having trouble following all of it. So I'm going to try really hard to keep it at a level that's understandable, to explain it where need be. I have not even begun to plumb the depths of all the different things that they talked about in these articles. But I will put them all in the description so you can go out and check those out for yourself. Let's go ahead and see what we can find. These are the 3 papers that I'm going to be talking about today. And I chose them partly because they are international, really. They're from all over the world, as you can see as we go along here. This first paper is from a group of researchers in Greece, Italy, and the United Kingdom. And I put the reference there for you to look at your leisure. The second one is from a group of researchers out of the Netherlands. And then finally, the third one is from a group of researchers in Spain. You can see that this is something that's gotten a lot of attention. So there are a couple of sections to the video that I'm making today. The first will be really to describe how it might work. What mechanisms might underlie the aging process at the cellular level. And then after that, I'm going to talk about what possibilities there might be for dealing with that. Now that we've learned some things, is there any application that we might make to help us with our aging or with reversing or halting multiple sclerosis? What I'm going to be doing all the way through here is just reading you quotations from the papers, and I will try to explain any concepts that I think are not easily understood. This first one is from the Oost et al. Paper, and they say cellular senescence, one of the major hallmarks of the aging process, is a phenomenon by which cells go into irreversible growth arrest and become resistant to apoptosis. Okay, so there's a couple of words right in there that you're probably scratching your head over. Senescence is basically just a fancy word for aging. We get our word senile from that same root. It has to do with old age. So the old age of cells is what we're talking about. So when cells get old, then we get old, basically. The mechanism involved is that cells, which, as we know, through their lifetime can replicate by division or other mechanisms, will go into some kind of irreversible growth arrest where they no longer can replicate. They become resistant also to what they call apoptosis, which is kind of a fun word to say. It just means planned cell death. So in other words, not death that happens because of some injury or illness. Obviously, you don't have every cell in your body that you were born with, and that was by planning. Cells get old and they need to retire and they need to pass away so new cells can take their place and do their jobs. And apoptosis is the process by which that happens in a planned way. And according to Ust et al., the number of senescent cells present in the human body increases with age, and that makes sense, which can have deleterious or harmful effects on the tissue microenvironment. So at the cellular level, we can see the harmful effects. And then down here, Papadopoulos has this to say: epidemiological evidence indicates that age is the strongest predictor for the transition from the relapsing phase of multiple sclerosis, which is considered primarily inflammatory, to the secondary progressive phase, which is mainly neurodegenerative. And I think you've heard that in other places, right? That in the relapsing remitting phase, we're mostly dealing with inflammation that comes and goes mostly. And that's why we get over our symptoms and go into some kind of remission for sometimes quite a long period of time. In the secondary progressive phase, the damage is more permanent and cumulative, and at that point we would call it a neurodegenerative disease. And as Papadopoulos and his co-workers say, although our understanding of the biological basis of aging remains incomplete, the prevailing hypothesis postulates that it is driven by the accumulation of irreparable molecular and cellular damage. And I think the important thing to note here is that you will see all the way through this, these are all hypotheses. There is not a clear answer to some of these questions, but the studies that they've done in humans and in mice seem to indicate that these are the mechanisms that are at work. So just keep that in mind. Maybe 10 years from now, they'll have a different idea. But this is what they're thinking now. Damaged senescent cells remain viable, so in other words, they are alive still, and metabolically active, so they still perform tasks. They accumulate with aging, and evidence suggests that their buildup may promote neurodegeneration. And then Dema et al. Had this to say: several studies suggest that autoimmunity itself, as well as multiple sclerosis progression, could be associated with premature immunosenescence. So in other words, the aging of our immune systems. Now I want to look at some of the mechanisms that they discuss. And the first one, and the big one, I think we all know, is inflammation. And here's what Ust et al. Had to say about that. Senescent cells may play a role in chronic tissue inflammation associated with the development of age-related diseases. Senescent cells accumulate in tissues manifesting age-related inflammatory pathologies, so in other words, pathology is disease, and promote this inflammation through pro-inflammatory factors, which may induce senescence in neighboring cells. So in other words, it's not bad enough that one cell gets this thing going, but it can secrete chemicals that get other cells in the neighborhood to do the same thing, sustaining and also exacerbating, making it worse, the mechanisms of tissue deterioration by increasing the number of senescent cells. So that's kind of a logical conclusion, right? If you're corrupting your whole neighborhood, then there's just more of you. And increased levels of pro-inflammatory molecules secreted by senescent cells can promote inflammation and might promote progression of MS. And Dema et al. Go on to say, One of the most important features of immunosenescence is inflammaging. That's a combination of inflammation and aging. A chronic low-grade inflammation, which results from an imbalance between pro-inflammatory and anti-inflammatory networks during aging. The principal stimulus involved in the progression of inflammation is the accumulation of cell debris. This is a big deal, by the way. Cell debris and self-antigens. So in other words, the things that would cause your body to attack itself, derived from cellular stress or viral infections. Hold on to that thought, because that'll come up again. The mechanisms devoted to cleaning cell debris and misfolded self-antigens become defective with age, leading to an overexposure of the immune system to these products and sustained low-grade inflammation, which favors autoimmunity. I think in a sense you've heard all this before, right? Now we're just looking at it more from the cellular level that goes on in your body all the time without you even really being aware of it. Another important factor is remyelination. And according to Ost and coworkers, in addition to suppressing of ongoing inflammation, remyelination is essential in MS to restore saltatory conduction and axonal protection. Promotion of remyelination and or inhibition of demyelination is critical to prevent further neuronal loss and cognitive decline observed in progressive MS. So neuronal loss just means loss of neurons, just in case that wasn't already clear. Failure of remyelination is one of the pathologic hallmarks of progressive MS. The relation of aging with disease progression in MS lends strong support to the hypothesis that progression could potentially be induced by increased cellular senescence in the central nervous system. Ost and coworkers also say the age-associated delay in remyelination can be explained by the decreased ability to resolve the inflammatory response initiated after myelin damage. So normally in the body, when there's been damage to the myelin, that your body immediately sends cells there to fix the problem. But as we age, or as our disease progresses, we get less and less able to respond in that way. Our body just doesn't work the way it used to work. The next thing to talk about is the idea of shortened telomeres. You may have heard about this. What I've got here is a picture of a chromosome. So this is what your chromosomes look like. Yeah, right. They're like little X's, and they have tons of DNA in there. And the human genome has been decoded recently, and we now know what all of the sequences are for the human and for many other organisms. But what you may or may not be aware of is that each strand of DNA has sort of a cap on the end of it, and they've shown it here as these browned ends. And these caps contain a lot of the same amino acids, but they are not actual DNA. They don't actually code for anything. What they do is keep the DNA strand from unraveling. But these telomere caps they don't last forever. Apparently every time a cell replicates, a little bit of telomere material is worn away, so that eventually the caps are pretty well deteriorated and the DNA strands can no longer hold together. And at that point, the cell or the DNA in that cell needs to just retire. Hopefully by then, the information has been passed into other cells that carry on the DNA coding, and that's not a big problem. But according to Papadopoulos et al., recently telomere length of white blood cells used as a measure of white blood cell biological age was found decreased in progressive multiple sclerosis patients compared to age-matched controls. However, shorter telomere lengths correlated with greater brain atrophy and higher levels of disability. suggesting that biological aging contributes to neuroaxonal loss and disability progression in multiple sclerosis. Total brain atrophy, as assessed by MRI, reflects primarily gray matter atrophy due to neurodegeneration and correlates with long-term disability in multiple sclerosis. So the shortening of the telomeres actually does directly correlate with our MS progression. And then according to Dema and co-authors, the aging of stem cells, HSC, that's blood stem cells, is characterized by impaired telomerase activity. A telomerase is an enzyme that increases the length of the telomeres or keeps them from getting shorter. So this leads to telomere shortening because the impaired activity of the telomerase causes the telomeres to shorten with each division. And as a consequence, the pool of stem cells is progressively reduced due to decreased self-renewal capacity. Seems kind of inevitable. And then the telomere length reduction and increased oxidative stress have been suggested as premature aging markers in many autoimmune diseases, including MS. The shortened telomeres and oxidative stress found in primary progressive MS patients may reflect the most severe state of the disease and may also be associated with greater disability and brain atrophy. Again, that's Dema and co-authors. Again, a hypothesis, but an intriguing one. Another factor that we would want to look at is the blood-brain barrier. And according to Papadopoulos et al., endothelial cell senescence. So endothelial cells are the ones that line the inside of your blood vessels. And when those get old, it may compromise the blood-brain barrier integrity, which is essential for preserving brain tissue homeostasis. And you remember, I'm sure you've heard other things about the blood-brain barrier, that when that is breached and stuff gets across it that shouldn't, that will threaten brain tissue. And again, from Oost et al., the senescence of endothelial cells lining the surface of blood vessels in the central nervous system is thought to contribute to disruption of blood-brain barrier function. And then there's a hypothesis about viral infection. I told you that this would come up again, so here's what's being thought about that. Dema and co-authors are saying that the main common characteristic between aging and multiple sclerosis is the accumulation of CD4 plus CD28 T cells. It's a kind of a T cell. These effector memory T cells result from repeated viral antigenic stimulation, typically CMV and EBV. CMV stands for cytomegalovirus, and EBV, of course, is Epstein-Barr virus. The interesting thing is that both of these viruses are in the herpes family of viruses, and once you get them, and they're very common apparently in the population, once you've been exposed to them, they stay in your system for your whole life. So in other words, these memory T cells result from repeated viral antigenic stimulation before migrating to the central nervous system, which contributes to tissue damage, thereby causing the release of self-antigens. It's postulated that these antibodies that we make in response to exposure to CMV or EBV may actually resemble too much of the stuff that the myelin is made from, and that may be why the myelin's being attacked. And a strategy to counteract immunosenescence is to reduce as much as possible the antigenic load represented by common infectious agents such as influenza virus and CMV to avoid inflammaging. And again, that's Dema and her coworkers. Okay, so now the second part of this talk, we're going to discuss what might be done with all this knowledge, or at least tantalizing suggestions that we have obtained in looking at cell senescence. And again, because all of what we think we've learned is hypothetical, what might be done is probably hypothetical times 2, so, or more than that. So just keep that in mind. But again, this is also interesting. I just felt like we needed to at least look at it. And I'll just read this. This is from Papadopoulos and his co-authors. Anti-senescent or senotherapeutic approaches may involve the selective death of senescent cells, senolysis is what that's called, to reduce the load of senescent cells and their detrimental effects on tissues. Senotherapy in multiple sclerosis would aim at preventing senescence-associated chronic inflammation, loss of cell function, and neuroaxonal loss, and promoting remyelination. So in other words, growing the myelin sheath back, which is sort of been a holy grail for MS researchers for quite some time. And Ust go on to say, potential therapeutic strategies to prevent the deleterious or harmful effects caused by senescent cells are based on preventing formation of senescent cells, removal of senescent cells, and targeting the effects of senescent cells. So there's 3 different options there. You can either prevent them forming in the first place, remove them once they're formed, or targeting their effects. And they go on to say, elimination of senescent cells can be done intermittently, which does not affect the formation of new senescent cells for purposes such as wound healing. And I think we need to talk about that for a second, because I haven't really mentioned that. Not all that is to do with senescence is bad as far as our health goes. Senescent cells, old cells, are often used in wound healing. And I can't really explain that mechanism to you here, but if you're interested, you can look that up. Maybe I'll find a resource I can put down in the description. And then you can see that if you target the cells that you want to get rid of and keep certain ones, this may allow for all the beneficial effects of having some senescent cells around without the downside that we're running into with aging and with progression in multiple sclerosis. So as they say here, different agents, including small molecules, peptides, and antibodies called senolytics, are being developed to specifically remove senescent cells. And then finally, a quote from Dema and co-authors. Rejuvenating the immune system could reverse the process of immunosenescence and have a beneficial impact on the treatment of multiple sclerosis as potential add-on treatments to current FDA-approved therapies. So in other words, if they come out with a way to eliminate certain senescent cells, that doesn't mean that you can quit your MS therapy. And they're very quick to point that out. Moreover, therapies focused on the rejuvenation of the immune system could provide new treatment opportunities for the primary progressive form of the disease. And then finally, Papadopoulos have the last word here. If senescent cells are proven to drive neurodegeneration, senotherapy may become the groundbreaking neuroprotective strategy to prevent and potentially reverse progressive disability in multiple sclerosis. And I know that you, like me, would love to see that happen. And since this video has gotten kind of long, I think I'll just stop there. But I'll do another video pretty soon where I talk about some of the senolytic senotherapies that are being looked at right now. There are a number of clinical trials that are in later phases even. So I think things may be coming into the approval phase pretty soon. But either way, I know you want to know. I know you're probably curious by now. What are we talking about? What kinds of things are we talking about? And I did say early on that some of them are prescription only and some of them are things you can buy over the counter. So I will let you know. I'll do another video and let you know. But leave me a comment. Let me know if there's something you already have heard about that you want me to talk about, or something that you want me to, a site that you want me to look at, whatever, a resource. I would love to know. So please keep me posted in the comments. Well, I hope that you found that all as interesting as I have been finding it. I will continue to look into this and to follow it. Some of the therapies that they're talking about are things that we might even be taking as supplements now, and other ones would need to be administered only under a doctor's supervision. But either way, I'm just impressed that they've learned so much and that they've made so many strides toward understanding why we age at the cellular level. What are the systems that are causing our bodies to slow down and fail? And how might those mechanisms be reversed? It's a fascinating topic. But that's all I have to say for now. In the meantime, until my next video, take care, and I'll see you again soon.